Now, on to the next kind of mechanism of action. Now, we have the PMOs, which are modified RNA-like AOs. Again, we have another very long name, which is basically your PMO, and it is also synthetic. So this morpholino analog, and it's stable and it's not metabolized with no apparent adverse effects, which is good. Now, these are basically antisense sequences which are about 25 bases long, and they target near your translational start sites, which is your ATG. And they have a very specific blocking of translation. Now, in terms of applications of your PMOs, they can be used as antivirals, whereby they downregulate the expression of your viral genes, like for example in Ebola. And this is effective against 75% of all your human viruses. The second application of your PMOs would be gene expression studies. So these can downregulate the expression of your study target. And a common term that you hear is the knocking out of genes. If you're having some trouble understanding this concept, you can think of it in the context of boxing. So in boxing, when you knock out an opponent, they are basically on the floor, unable to do anything. So I think it helps when you have an example in mind. So let me just play a short video here for you. This is the zebrafish which have not been knocked out. So you can see they are very responsive to the pipette tip. Whereas again, they are being prodded, but as you can see, they're not really moving. Well, that one moved, but most of them are not moving that much as compared to here, where you see they just, the moment they're touched with it, they just scoot off. Okay, now we're going to do a bit of revision from Dr. Frigal's lecture. So, your DMD is an X-linked recessive disorder, and it only affects boys. So, because it's X-linked recessive, and males only have one X chromosome. And you have it, well, it's a muscle wasting disorder, as you know, and it can be due to frame shifting or nonsense mutation in your dystrophin gene, which results in protein truncation. And it's very progressive, so ultimately it results in respiratory or cardiac failure. So again, I think these examples and clinical cases help to cement this into your memory. So if you think of the poor boys who died when their ventilators failed during a power outage, you can imagine just how severe the DMD can progress to such a state. And your DMD carriers, on the other hand, have about they have dystrophin in about 50% of their muscle fibers. So just make note of the size of your dystrophin gene. Yeah. Okay, with DMD versus your BMD, which is your Becker muscular dystrophy, which is a less severe phenotype, it's due to in-frame mutations, and with DMD, it's because of mutations in these two regions. Whereas in BMD, where it is a less severe phenotype, it's in these regions here. Now, Dr. Frigal has a very nice graphic here, so if you're a visual kind of learner, that's for you. Okay, now, gene therapies for DMD. We have your mini dystrophin gene delivery. So you basically have smaller dystrophin genes which are delivered via plasmid and viral vectors. Or you have a transcriptional read-through. So these are delivered in the form of antibiotics, which cause your ribosomes to ignore your stop codons. So that where there's not meant to be a stop codon, there is. And when you give this antibiotic, it ignores the stop codon. So you don't actually get your protein truncation. Now, another thing that you can do is your homolog induction. So this dystrophin analog, utrophin, is similar to dystrophin, but it's, if I'm not wrong, it's the fetal form of dystrophin. So it has a promoter which may allow your selective upregulation. And another thing is your cell-based whole gene replacement, which is Dr. Fugawa's research project. And here we have, you basically inject donor myoblasts, which have a functional dystrophin gene, into your recipient, and this procedure is called your myoblast transfer therapy. And here I've got a nice little handy table as to what Clayton used for various components of his study. Okay, and finally we are on to exon skipping, which is Professor Fletcher's very fantastic research. Again, I just reiterate the different names for her developed therapy, which is, that's right, your antisense oligonucleotides. And it works by skipping your exons because you have a stop codon where it's not supposed to be, and if you skip that exon, you skip the problem. And this induces a Becker-like mRNA in vivo, and just if there's nothing you understand from this sub-review, just know, okay, that it's very important to know that some dystrophin is better than no dystrophin. Okay, now the PMO, you can refer back to slide 10 if you've forgotten what that is. It targets the dystrophin exon 51, and it prevents the selection of the exon during splicing, which results in exon skipping. And this is successful in 13% of your DMD patients. Now, with the process of Professor Fletcher's research, she had a primary endpoint, which was ambulation, and the test which they used was a 6-minute walk test. She's got a very nice graph, so refer to that. And the secondary endpoint, the secondary endpoint was the amount of dystrophin expressed. So you can see that at the end, after they administered the exon D51, there was an 11-fold increase, which you can see here and here, that there is more green fluorescence, but it's still low expression compared to normal. Again, let me just reiterate this so that you remember it. Some dystrophin is better than none. Alright, now, in the last few slides of Professor Fletcher's lecture, she went through SMA, and this is a homozygous loss of SMN on chromosome 5, and this is its size. Now, there's a near identical gene called SMN2, and this differs by 5 base changes. But the problem with this is that it weakens the selection for exon 7, so you end up with a decreased SMN full-length mRNA. And there are 2 types of SMA. SMA1, type 1, is worse than SMA2. And you can see here that if you have greater copies of SMN2, you have a decreased severity of SMA. Okay, now to treat SMA, again, we've got a very long name which I'm not going to pronounce right, so I'm not going to say it out. And because if you remember, with SMA, you get a reduced selection for exon 7, which is a problem. So what this therapy does is it increases the inclusion of exon 7 in SMN2. And this therapy here is what Professor Fletcher has developed, but there is another therapy that is already in clinical trials called this, and it's been very successful. So it's kind of disruptive for the further development of other oligotherapies because it works so well. And I think they're administering this IV, but at this stage it's unknown if they need multiple rounds of treatment. And a problem that could arise from that with retreatment is that the patient can acquire immunity to the adeno-associated vector, which is this. Okay, so that was my last slide. These are just some references, and you can look them up if you'd like to. If you're interested as to reading a more in-depth paper as to what exon 51 is about, that's the link right there. And